Welcome everyone to this morning's webinar, COVID-19 in Chemistry. My name is Sarah Mullins and I'm your host for this morning's webinar, and I hope that together we can learn a bit more about research our fellow scientists are doing in the COVID-19 space. Before we go further, I acknowledge the traditional custodians of the land that we are standing on and pay my respects to their elders past, present and emerging. I would like to extend that respect to Aboriginals and Torres Strait Islanders that join us today and recognise their contributions as Australia's first chemists and engineers. This morning, I would like to welcome Dr. Kirsty Short, Australian Research Fellow at the University of Queensland School of Chemistry and Molecular Biosciences, and Dr. Paul Birch, Science Director for Land and Water at CSIRO. Firstly, please welcome Dr. Kirsty Short.
Thank you very much. All right, let me just navigate the screen sharing. I think by 2021 I'd have this down pat, but never mind. Sorry. All right, can everyone see and hear me? Yep, I'm going with a yes, otherwise yell out if you can't. All right, so it's my absolute pleasure to be here today to talk to you a little bit about the research that my group has been doing during this SARS-CoV-2 outbreak or the COVID-19 pandemic. So we're a virology group, certainly not chemistry, and I'm certainly not engineering, but don't hold that against me. So what we're particularly interested in, even long before the COVID-19 pandemic, was emerging respiratory viruses and sort of pandemic preparedness. Because as shocking as the COVID-19 pandemic has seemed, this is something that was going to happen. We just didn't know which virus and when. So one of the things that struck us in the initial outbreak of the pandemic was really the role of children and trying to understand what the role of children in the transmission of this virus was. So we've worked for many years on influenza, and we've known that children really drive the spread of influenza in the community. This is one of the major reasons that children are advised to be vaccinated against influenza. It's not just to protect themselves, but it's also that by vaccinating children, you can protect the broader community. But the role of children and the spread of SARS-CoV-2 is much more controversial, and this is certainly an area that was sort of highly politicized early on in the pandemic. And what we really wanted to know is what does the data say? What do we know actually about the role of children spreading this virus? So to do this, what we did is we performed some epidemiology studies looking at household transmission clusters of SARS-CoV-2. So when you had the virus infecting a household and it spread within that household. And what we wanted to know is that when there was a household transmission cluster, how often was a child in the family the index case? Or how often was it that the child was the first one to bring the virus into the home? And so we gathered this data. Again, this was mainly in the early stages of the pandemic across every country where the data was available, where these household transmission clusters were defined. And so when we put this data together, what we found is we were able to identify 213 family clusters of SARS-CoV-2 infection. And of these 213, 8 had the child as the index case of the cluster. So only in 8 clusters was it the child bringing the virus into the home. And in all the remaining clusters, it was the adult in the family who was infected initially. So this would suggest that probably children aren't really driving the transmission of SARS-CoV-2. But there's a lot of caveats to this data that you've got to take into account. So the first thing is that children are often mildly symptomatic with SARS-CoV-2, or they can have asymptomatic disease. So there was a possibility that what was happening was that the child was actually the first one bringing in the virus home, but they were just not being identified because they were asymptomatic. So we went through all the data and we looked at clusters where there was an asymptomatic child. And in those clusters, we just assumed it was the child bringing the virus into the home. And even if we did that, that brought our number of child clusters up to 39, but still the overwhelming number of cases, it was the adult bringing it into the home. Okay, well, you may say that because we were looking at the early stages of the pandemic, a lot of the cases of infection were associated with travel to endemic areas, in particular in China, travel to Wuhan and more generally Hubei province. So what we did is we took out all cases which involved travel, because what you've got to remember is that children are not going to travel outside of the home independently, whereas adults will. So we looked at that. And again, even if you took this out of the equation, you still saw that children were in the minority of transmission clusters. So again, you could say, well, this is probably because this all occurred at a time when schools were closed, so children weren't getting infected as easy. But again, we went through the data and only looked at circumstances where schools were open. And again, even when schools were open, children were still in the minority of the number of index cases. And just to give you a point of comparison, what you can see is that if you do this same study with influenza, you see very different results. So this is a study of the household transmission clusters of avian influenza. And what you can see here is that actually children are the index case in the majority of cases. So this to us is really suggesting that there's a fundamental difference in the role of children in the spread of SARS-CoV-2 and influenza. Now, because we have this data, we could also look at within the family, how likely was a child to be infected? So independent of who brought the virus home into the family, if there was virus in the family, how likely was a child to acquire the infection versus an adult acquiring the infection? And I'm not going to go into too much detail here and go through all these forest plots, but I'm just going to say that children were significantly less likely than adults to acquire the infection in the home, although we did not see a difference between younger and older children. So children under 10 looked much the same as children over 10. So if we put this all in context, what it's suggesting is that if a child is exposed or infected with the virus, it leads to either no infection or rapid resolution. As such, the child does not bring the virus home into the family. If you have a context where an adult is infected and is infected within the family, then a child either rapidly clears the infection or is not infected. So these two scenarios really creates the question, why would children be more resistant to infection? Now, there's lots of different theories about this. Some suggest it's about pre-existing antibodies to seasonal coronaviruses. But what we wanted to know is there something fundamentally different in the antiviral response of pediatric nasal epithelial cells compared to those of adults? And the reason we're focused on these nasal epithelial cells is they're really the first site of virus replication. So they're the sort of first barrier that your body has against infection. So to answer this question, we did nasal samples from adults and pediatric patients, and we cultured these epithelial cells that we took out of the nose at an air-liquid interface, so mimicking what happens in the nose where these cells are exposed to air, and then we infected them with SARS-CoV-2. So prior to infection, when you look at how these cells grow in a culture, you can see that the adult cells actually look quite different to the pediatric cells. And specifically, the pediatric cells have these pseudocysts, which is consistent with a cell that has a sort of elevated metabolic activity, which is what we think is happening in the pediatric cells. When you just look at the basal gene expression of these cells, what you can also see is that the pediatric cells and the adult cells look very different. And this is why they're clustering different on this PCA plot. And one of the things that's really interesting is that the pediatric cells relative to the adult cells have an upregulated interferon alpha response. Now, this interferon alpha response is, of course, associated with resistance to viral infection. So even just at a basal level prior to infection, we're seeing that the pediatric cells are probably growing a bit better. They're a bit more metabolically active, and they have a higher basal interferon response, so a basal antiviral response. Now, there's been some suggestions that one of the reasons that kids might be less susceptible to SARS-CoV-2 is that they express different levels of the viral receptors for SARS-CoV-2, specifically the ACE2 receptor and the TMPRSS2 protease. So we actually looked at this in our pediatric and adult cells, and we didn't see any significant difference in the protein level expression of either ACE2 or TMPRSS2 between our adult and our pediatric donors. So this to us suggested, well, maybe there's something else going on with these nasal cells. So we infected these cells with SARS-CoV-2 and we looked at the replication of the virus. And what you can see here is really, really striking. Our adult cells are in blue and our pediatric cells are in pink. And when you look at the replication of SARS-CoV-2, what you can see is that it replicates infinitely better in the adult cells compared to the pediatric cells. You can see this same thing by Western blot as well. So if you look down here at the Western blot, this is for a viral protein. This is the adult cells at 72 hours post-infection, really nice thick band. And this is the pediatric cells. So there's this inherent difference in how the virus replicates. You can then do these experiments with a different virus. So if you remember, I told you that influenza virus is really the transmission of influenza virus in the communities driven by children. And what you can see is that with influenza virus, the virus, if anything, replicates better in the pediatric cells compared to the adult cells. So there is something about SARS-CoV-2 that lets it replicate better in the adult cells. We then did a whole bunch of transcriptomics to try and figure this out. And I'm not going to go into too much detail about this. All I'm going to say is that the differential gene expression that we saw between the infected pediatric and the infected adult cells was really, again, in this antiviral response. And we saw that the pediatric cells were able to mount this much stronger, more robust antiviral response compared to the adult cells. And that sort of makes sense when you start thinking about those replication differences I showed you. Now you can see these same data by qPCR if we look at some interferon regulated genes. And what you can see is that at 48 hours post-infection, they're much higher upregulated in the pediatric cells compared to the adult cells. What's interesting is if you do these same experiments with influenza, you also see that in response to influenza, the pediatric cells mount a stronger antiviral response. So the question is then, if there is something fundamental about these pediatric nasal epithelial cells that's making them launch a better response, a better antiviral response, why is it working against SARS-CoV-2 and not influenza? Well, we know that actually influenza is much less susceptible to these type 1 interferon genes compared to SARS-CoV-2. So what we think is happening here is that the pediatric nasal epithelial cells have a higher interferon response. This is sufficient to control SARS-CoV-2 replication, and as a result, kids aren't driving the transmission of this virus, but it's not sufficient to control the replication of influenza virus, and as a result, kids play a much more important role in the transmission of influenza in the community. So that's sort of a brief snapshot as to how we've been keeping ourselves occupied during this pandemic. And really, I think the sort of take-home message is here is that at least in the early stage of the pandemic, and there's lots of debate going on about what's happening now, is that children are really unlikely to be driving the household transmission of SARS-CoV-2, and that they're fundamentally less likely to be infected than adults. And this probably reflects, at least on some level, fundamental differences in the nasal epithelial cells of children versus adults. So thank you very much. I'm happy to take any questions because I'm aware that's a lot of information to churn through and no chemistry whatsoever. So thank you very much for being patient with me. Are there any questions?
If anyone has any questions, they can enter them into the chat or into the Q&A box down below.
Stunned everyone into silence. All good.
It was a good talk. I thought it was very clear. Epithelial cells and interferon genes. So it was a good talk. Maybe you've just been very clear.
Yeah, I mean, one of the things that we're doing at the moment in the lab is we're looking to see if these same trends hold true with the new variants, because there's been a lot of discussion, especially in the pediatric community, that maybe these variants have a propensity to infect children over adults. So we're trying that right now with the UK variant, and we'll try it shortly with the South African variant as well.
Oh, that's good. And no results just yet.
Literally, the cells are incubating, so we'll get the results in 2 days. I'm super excited. Yeah, I'm super excited. I don't mind which way it goes. I just want to know.
Yes, look, that's fair. Okay, well, if there's no more questions at this time, we can have, and if anyone thinks of anything at the end, we can come back to questions. Otherwise, thank you, Kirsty. And please welcome Dr. Paul Birch.
Thank you, Sarah. Here we go. It seems it's saying my screen sharing is paused. Of course, I did it earlier.
I'm not sure.
I'm not
Pam,
sure what that
do you know
means.
how to? I'm not sure what that means either.
No. Let me stop share and start again.
Okay.
See what happens there.
Do you just need to press play on the PowerPoint?
Yeah, there we go.
Lovely.
Thank you. Not sure what happened there, the pausing of my screen share, but yeah, thank you very much. And what I'm going to be discussing is wastewater-based epidemiology for surveillance of COVID-19 in communities. And this is work that has arisen very quickly globally as a result of the pandemic. But I should say that wastewater-based epidemiology for both chemicals in wastewater and biologics have been used for decades. And here in Australia, not so much for biologics, so it's mainly been chemistry. And our colleagues at the University of Queensland run the National Drug Monitoring Program for the ACIC, the Australian Criminal Investigation Commission. And what they do is look for illicit drugs as well as alcohol, tobacco, et cetera. But you can essentially measure anything in wastewater and it's an integration of things that we actually are exposed to either through ingestion or incidentally. And I have this image here that just came out, and we are also looking at being able to detect the variants in wastewater currently. But let me start from the beginning. Warsh Ahmed is the scientist at CSIRO who leads up this work. And then the Queensland Alliance for Environmental Health Science, our colleagues, Professor Thomas and Mueller, have been involved with this as well. And Warsh was already participating in an international collaboration prior to being contacted by Professor Thomas and Mueller to see if he could actually measure the SARS-CoV-2 genes in wastewater. I should say that Queensland government and Queensland Health has been funding most of this work, and Greg Jackson is the individual that looks after that for Queensland Health. And we've also been getting support from Qantas because we've been doing long haul flights for Qantas. And we've also have done cruise ships, as I'll talk about in a moment. But just looking at the team here in Brisbane, we're based at the Ecosciences Precinct at Dutton Park, which is just across the railroad tracks actually from the Queensland Alliance for Environmental Health Science. And in the near future, when the Crossover Rail project is completed, we'll actually have a bridge that we can walk across, because right now it takes quite a bit of time to go around and underneath the tracks. This is just an infographic that summarizes really what we're looking at, and then I'll get into more of the specifics later on. But what basically happens is when an individual becomes infected, within just a few days, they typically shed the virus in their feces. Kirsty has already mentioned ACE2 receptors. ACE2 receptors are very specific, or I should say the virus is very specific for those receptors. And those ACE2 receptors are actually highly concentrated in intestinal tissues, and thus coronaviruses generally, so SARS-CoV-1 was also shed quite early. So this would typically be an individual that's infected would typically be shedding the virus before they would show symptoms. And the beauty is, even if they're so that's pre-symptomatic, but even if they're asymptomatic, they shed the virus in their feces. So the samples can be collected, and within a day, then the gene fragments can be determined to be either present or absent, and one can actually get some estimates of the number of infected individuals also in a catchment. And I should just say that the detection and quantification of the virus materials then can be reported to the public health officials. And here in Australia, Queensland, Victoria, and New South Wales are regularly posting data from their wastewater-based epidemiology surveillance for SARS-CoV-2 regularly. Other states, other jurisdictions are tending not to post that data, but are collecting that data as well. So in March, there was a global race in terms of trying to understand trying to understand the ability to actually measure gene fragments from SARS-CoV-2 in wastewater. The UQ-CSIRO collaboration commenced in March with interest from the Australian Criminal Intelligence Commission. And again, Warish Ahmed led the first study on wastewater-based analysis. I should say that there was quickly work going on around the world. And as Kirsty would know, and many others that are working in COVID-19 research would know, there was a number of, or unprecedented number of papers being posted that had not been peer reviewed yet, and which oftentimes was a bit challenging. But early studies that were posted and not peer reviewed from the Netherlands, France, and Spain, and the US all indicated that detection of the virus in wastewater preceded any detection of individuals that were actually infected in the community via the nasopharyngeal methods. And Warish's paper with his colleagues then was the first peer review paper in the world, so which demonstrated that here, obviously in Australia. And this data here just shows this is in wastewater versus the clinical test, and you can see the signal in the wastewater precedes that. And in the US, a study by Harvard and MIT, along with a company called BioBot, which is a startup company in wastewater-based epidemiology that actually started up to look at opioids around the opioids crisis in the US, but they pivoted and now are servicing about 700 wastewater treatment plants across the US twice weekly and bringing in about 9 million a year doing that. And so this is catching on very quickly. Again, just that first paper actually, and I should say that I started out as a spokesperson for CSIRO and then became involved in the study after looking at the first paper because I had some ideas around methodology that could enhance the sensitivity of the technique, and I'll be talking about that in a moment. But these are just some positives that were showing up again, and these are clinical cases here. And I'll talk about this a little bit later as well in terms of some of the data that was, again, showing up in advance of clinical determinations of infections in the community. So one of the things that was also done was to go back and look at samples that had been collected and stored, but in advance of actual sampling. And this, again, is showing different positive samples. These red bars are positive samples. The first positive sample was in late January. at a wastewater treatment plant in north of the river in Brisbane. And this was not actually a big surprise in that we know that there were 2 individuals that had disembarked in Darwin on the Ruby Princess and then traveled to Brisbane. And one of them was in hospital. And so we were detecting the viral fragments from wastewater of, in this case, one individual, and that catchment has about 300,000 individuals there. So you can see it's a quite sensitive technique, but I can talk a little bit about some of the challenges, analytical challenges anyway, going forward. One of the really interesting things, though, was in a wastewater treatment plant in the southern part of Brisbane, and I'll talk about that a little bit later in terms of the early warning system, but we were actually detecting positives about 3 weeks, 2 to 3 weeks before the first clinical diagnosis for an individual in the south part of Brisbane. So again, that early warning detection is what's very, very important here, particularly in areas where there's low incidence of disease, such as Australia. After that, there was a workshop to evaluate and refine methods that I was quite interested in. And again, this is looking at sampling methods and virus stability, looking at concentration extraction methods, RNA extraction methods, assessing different primers, as well as looking at when and why you would want to sequence. At the beginning, we were sequencing all of them just to confirm that our amplified products were in fact representative of the gene fragments that we had thought, and then also in terms of reporting. So the temporal and spatial trend monitoring program on the archive samples, I've already covered that, and also access to other applications. And we've done cruise ships, we've talked about naval ships, we've done airplanes and other specific assets. You can do prisons, and we have done aged care facilities, et cetera. So it's the level of resolution can be quite significant if one is interested in that. And I'll come back to that a little bit later, because globally there's a lot of interest in actually upscaling this and getting much higher resolution at the community level down to the postcode, for example, or individual dormitory. And then there's key uncertainties for quantitative use. One is the variability of viral shedding. That is, individuals vary quite significantly in their viral shedding. But as the studies in Boston in the US demonstrated, even the great variability in that viral shedding and the estimates that would range quite significantly are still much better than what they were actually finding from reporting from clinical positives. And so this was another paper looking at the comparison of the virus concentration methods. So this is one of the things that I was quite interested in by looking at those initial methods, thinking that we could optimize those methods for getting much greater efficiency of the virus being actually concentrated from the wastewater samples. So the 2 highlighted areas here, the method A was actually the method that was originally used. So it was only capturing about 26.7% of the viral fragments. And we were able to, through method optimization, get that up to closer to 66%. And further refinement now, we believe we will get even higher, closer to 80 to potentially even 90%. I should mention that these are the methods that we examined in this particular paper, but other methods that are still being used globally, and oftentimes you'll find that they're used quite frequently, are as low as 13% in terms of that recovery. So you can see that, and I'll talk about this because this is quite significant in terms of what people are concentrating on, and I'll get to this in a moment. But in terms of thinking about QA/QC and some of the workflows, oftentimes are focused really on the actual amplification of the genes and the detection, rather than further down through the whole sampling scheme. And this is quite significant. Another paper actually looked at the decay, and you'll see temperature is, of course, a very important variable. And the other thing that this paper looked at was to demonstrate that another coronavirus, which is the murine hepatitis A virus, could be used quite effectively as a surrogate for the SARS-CoV-2, which makes it much easier to work in the laboratory. And this just demonstrates, again, temperature being a very important variable in terms of the degradation. But under the conditions here in Australia, even temperature isn't necessarily a problem in terms of the number of days, given the actual architecture of most of our wastewater treatment plants, even in places like Darwin, where you might expect quite high temperatures. Untreated wastewater, and obviously autoclaved wastewater was a little bit better. I cut out the work in terms of the tap water, but obviously the lifetime in tap water was greater than autoclaved wastewater, and certainly than untreated wastewater. So there's a number of things that would break down the virus. Certainly there are a number of detergents in wastewater, and this particular virus is very sensitive to detergents. And again, this particular study was actually looking at the sampling scheme. And this is really critical because very few people have concentrated on this at all. And if you look globally at most of the studies that are being conducted, and even the routine analyses now, most studies and most monitoring systems are just taking simple grab samples. And what this study actually looked at was the difference between grab samples, 1-hour composite samples, and 24-hour composite samples. And what we found was that the 24-hour composite samples were much more consistent, and also much more sensitive than the 1-hour, and certainly much more so than just a simple grab sample. And this is not really a big surprise, in that if you have chemicals that are being excreted, say in urine, at a relatively constant rates throughout the day, then you don't expect large peaks and valleys in terms of the actual signal. But with the viral fragments coming from feces, this can be a bit tricky in terms of really understanding when you would be taking that sample, given that the travel time can be up to 12 hours, or in some cases, depending on the architecture of the wastewater treatment system, could be up to potentially days. And there's a whole host of things relative to the hydraulics as well, in terms of mixing, et cetera, that also introduce variability into that. So this is what I wanted to again come back to, is if you look at a lot of what's going on in the world, and again, coming more from a clinical perspective, and this is the Centers for Disease Control in the US, they're really focusing on analysis after the sample is brought to the lab. So they're not even, the sample concentration, a lot of, again, as I mentioned, very few studies have looked at different concentration methods as we have. And so there's quite a bit of variability there. And really what we're suggesting is that you really have to think about the whole sampling, transport, and then lab chain to really understand the sensitivity, and understand false negatives and false positives in the wastewater. So, for example, many studies, it's really even hard to see, outside of taking a grab sample, what kind of standards that they're using, what kind of blanks that they're using, even in the transportation. And there's possibilities of contamination all along this chain. So it's quite important to consider that. And again, wastewater is, of course, quite a complex matrix. So there's a number of tricks that oftentimes individuals that are picking this work up, that are coming from more of a clinical background, there's a lot of nuances that are being missed, allowing for some variability there. And then this one was actually looking at optimization and quality control, again, as crucial for generating reliable public health information. And this is the scheme that we've worked up. Again, this is looking at the whole process control from actually sampling, transport, and then how do you actually process the samples in the laboratory. And a paper that has not yet been peer-reviewed, but it's now in preprints, is actually looking at a comprehensive review of all of the studies that have gone on globally, and tried to put that into a system to frame where the best practices may in fact be. So again, a lot of focus on the assay limit of detection or quantification rather than the method limit of detection, which really looks across that whole chain. And one of the things about our true positives and false positives is we believe false positives, if with a good workflow and proper QA/QC, can be controlled. The real problem that we see really is false negatives due to the lack of sensitivity by not using best practices in terms of actual concentrating and while handling the samples and then concentrating them properly. And then there's also differences in some of the primer and some of the genes areas that are targeted and again, oftentimes not accounted for in certain methods that, or certain studies, or even monitoring systems that are just using one. So again, Queensland Health has been doing a remarkable job in terms of rolling out their program. You may see that the Chief Health Officer oftentimes reports on the wastewater positives across the state. One of the things that she does quite well is to point out that really the positive, and so one thing that's called false positive sometime is that individuals continue to shed after they are believed not to be infectious. And so that can all go on for up to 2 months after they've recovered, which is an issue. I don't call that a false positive, because that's a true positive. It's just that it's believed that those individuals aren't infectious. China actually views it differently, and they actually require negative anal swabs prior to allowing individuals to go back into the community, because they're not convinced. Again, there's a lot of data that's not peer reviewed, but they're not convinced that those individuals couldn't still be infectious at some level, although the data that we're aware of would suggest that that's probably a very low probability. So the timeline, there was a pilot study that this should be October, but went from July 27 to October 26, and then routine monitoring started on the 27th of October, and it's continuing till June 30th. To date, over 2,000 samples have been run. I should say that there's been some challenges in terms of actually getting acceptance of wastewater-based epidemiology by the health community more broadly. At the beginning of the pandemic, a lot of clinicians were seeing this really as a distraction. They didn't understand the unique information that was being actual generated, and it's complementary. So at no point would we suggest that this would be a replacement, certainly for clinical testing. But certainly particularly for poor countries or countries that have difficult in accessing samples or clinical sampling, this is quite powerful for them as well. And the last thing I really wanted to touch upon was the international collaboration. And I suspect many of you have heard about the unprecedented international collaboration that has emerged around all aspects of the pandemic, but certainly in this area, it's been quite significant. There's a international collaborative, and you can see now they keep this data up to date. I just took this off the web this morning. There's currently 70 dashboards that they're aware of internationally. You can see the number of countries that are actually doing routine monitoring and the number of sites that are routinely posting their data are now over 2,000. So this is really accelerating and taking off in terms of being used as a tool more broadly across the world. So in conclusion, I just wanted to point out that wastewater-based epidemiology is a powerful, complementary, and unique approach for surveillance of wastewater for understanding community transmission of COVID-19. As I mentioned, it provides community-level information by aggregating individuals in the community, and it really has a high level of sensitivity. As I mentioned, we know that we can detect 1 in 300,000 individuals. There's been a desktop study that was conducted at the Arizona State University that suggests under the best case scenario that you would be expected to be able to detect 1 in 2 million, and under the worst case scenario, and I can talk about what those scenarios are in the questions and answers if people would like, but under worst case, it would be about 1 in 110,000. We've demonstrated, and others have as well, that this is a very powerful early warning detection system. You can actually detect the virus in a community before there is any evidence of clinical positives. This is very important because, as I mentioned earlier, asymptomatic individuals also shed the virus. And you may have seen that the Centers for Disease Control in the US just a few weeks ago, it estimated that 50 to 60% of all cases in the US at the time were being actually transmitted from asymptomatic individuals. So that's a very significant number. And it's been demonstrated pretty consistently across the world that between 30 and 60% of all cases are transmitted from asymptomatic individuals. The granularity can be increased to the catchment or postcode level, as well as I mentioned, individual asset. And so this is another area that really hasn't been pursued to the degree that it potentially could be yet, but I'll talk about it in terms of the global interest in the next, in the bullet after the next one. Consideration of QA, QC, and workflows need to be across the entire sampling, transportation, processing, and measurement chain. And this is, as I mentioned, has been a big problem. And we're concerned that potential to undermine the confidence in wastewater-based epidemiology by individuals that are conducting it that don't really have a full appreciation for the complexities in terms of this whole chain of sampling and processing. The global interest is growing, as I mentioned earlier, rapidly, but what's also growing is the desire for strategies for more permanent surveillance schemes being designed into new infrastructure. So for example, we were approached by the Asian Development Bank and asked to put a proposal in for infrastructure development for wastewater treatment systems in Indonesia, where they would have built in the ability to sample at a very high level of resolution. And that's also happening. We were just recently contacted by the Philippines for very similar projects. I do have colleagues in Singapore, and they already have that capability because they are very aggressive in their drug monitoring, their illicit drug monitoring at a very fine scale of resolution. And they're using that quite effectively as a wastewater-based epidemiological tool. So I will stop there and happy to take questions.
Thanks, Paul. Does anyone have any questions either in the chat or on the Q&A setting? Otherwise, I'll ask one while we're waiting. So you mentioned the resolution of that, that last point you made can get down to the dormitory level. How would that be possible? Is it just the sampling to the dormitory level? Yeah.
Yeah, it's just the sampling, that's right. And Singapore did go to the dormitory level because you may know their second wave actually came from foreign workers living in dormitories. And so they went to the dormitory level. They also have a lot of the time integration data coming out of those dorms. And so they just integrate between 5 and 10 a.m. every morning. So you don't have the mixing problems and some of the travel time issues, obviously, the further up pipe you go. The University of Arizona in the US, when they went back into session back in August, were doing dorm level sampling, and they had some big success stories early on where they had some positive detects on a dorm. They went in, they sampled all 316 students that were actually living in that dormitory. They found 2 asymptomatic cases. They were able to isolate them, and no other students were infected. And so that's been rolled out, from what I understand, pretty much across the US at universities. It's become pretty common to do dormitory scale measurement.
Okay. And you mentioned that assay limit of detection versus method limit of detection. What's the difference between those two?
Yeah, so again, the assay is just, what assays you're using in the lab. And so it's really looking at, what kinds of interferences there might be or not, and just really understanding that part of it. Whereas the method level of the detection starts all the way from how you sample. So if you take the grab sample and you take that grab sample, it happens to be not in sync with whatever the peak of the virus actually arriving at wherever it is you're sampling, then obviously you're going to miss it. So you could have certainly you could have positives in a catchment, and you're just missing it by just taking a grab sample. And we found, even with the 1 hour integrated sample, it was much greater variability and much less sensitivity than the 24-hour.
Interesting.
And then that would also be the concentration methods. As I mentioned, if you look at the methods used in around the world, there's still quite a few instances where you can see methods that are only 13% efficient at concentrating the virus. So again, that's 13 versus, as I mentioned, we're up to around 67. We're pushing that higher. And it's just really understanding what the surface chemistry of the virus is and how you can best concentrate that virus.
Okay, I've got a question.
Keeping in mind that it's quite small as well, right? Because 70 to 90 nanometer size range. A lot of the typical filters that are used are much larger than that. So it's really understanding.
Yes. I've got a question from Mary. How long does it take to analyze a sample? What's the turnaround time?
So the turnaround time is about 4 hours once the sample gets to the lab. And so the logistic problems, of course, is you have people sampling the wastewater, then you have the transport. But once it gets to the lab, it's about 4 hours turnaround time.
Okay.
We typically turn them around in a day for Queensland Health. Okay. Well, if we don't have any other questions, thank you, Paul. That was great. Good to hear that information. I'd also like to thank the Royal Australian Chemical Institute and the Analytical Environmental Division for supporting this webinar. And finally, thank you everyone for coming along and listening. If you have any further questions, please let me know. Otherwise, thank you everyone, and I hope you enjoy your day.